Уважаемые коллеги, я думаю, большинство присутствующих не случайным образом оказались в этом зале. Для меня огромное удовольствие и честь представить вам лекцию профессора Мишеля Саделайна из Sloan Kettering Memorial Cancer Center в Нью-Йорке. Я, пожалуй, без формального представления обойдусь. Мишель – это человек, который стоит за созданием одного из наиболее значительных прорывных направлений в современной медицине, в онкологии, адаптивная иммунотерапия, КАР-Т лимфоцитами. И помимо того, что он создал это направление, он сыграл большую роль в том, что нам удалось сделать в Центре с точки зрения перенесения технологии и применения ее у конкретных пациентов. В самом начале мы получили бесценные советы о том, каким путем нам лучше двигаться, и я надеюсь, что лучше, чем он сам, никто не расскажет о том, как это все началось и что нас ждет впереди. Michelle, you're welcome to present, and I hope there will be many questions.
Well, thank you, Professor Maschan. It's a tremendous pleasure for me to be here for the first time in Russia and to visit this beautiful center and to also discover how you are putting into place a program that builds on CAR T cells, Chimeric Antigen Receptor Engineered T cells. That happens to be one of my favorite topics. I think we can all agree that there are still today too few treatments in cancer that are curative. I insist on the word curative, meaning a treatment that can be administered ideally only once and can eliminate for good the cancer. We all know that a key feature to achieve a successful treatment is to reconcile safety and efficacy, and that the best way to do that is through the use of specific treatments that will specifically attack the cancer and cause minimal damage to normal tissues. It turns out that nature has given us a wonderful tool that is highly specific, and that's the adaptive immune system, chiefly composed of T lymphocytes. Not only are T lymphocytes highly specific, but they can also last. They can induce what is called immunological memory. Unfortunately, T cells don't always have the potency, the strength to kill the tumor cells or to function in the microenvironment that cancers create to protect themselves against immune attacks. And so many years ago, 25 years ago or so for me, we decided that we could help the immune system through genetic engineering, give instructions to the immune system to recognize and destroy cancer cells. In a very simple overview, the idea is to design T cells that have been genetically engineered to recognize the cancer, give them to the patient in the bloodstream, and allow for these T cells to now find the cancer and destroy them. The hope is that the destruction of these cancer cells will prime other cells in the body of the patients to continue the attack, but genetically engineering can also be used to augment the endogenous response to the cancer. So what I would like to discuss with you in this first part is something we call Cd19 Car Therapy. It is something that we started at my center many years ago that has then been embraced by the National Cancer Institute and the University of Pennsylvania in the United States, and then led to the approval of the first CAR therapies last year in the United States. This is an atypical field. It draws on multiple fields at the intersection of these fields. First, we learn a lot from immunology and transplantation, the principles of T cell biology, and today also teaching us the tumor microenvironment, how the tumor protects itself from immune attacks. So we need all of this basic knowledge. But then this field is uniquely built on the use of genetic engineering. I will discuss some of the tools later that we have to engineer the immune system. Another unique aspect of this field is the design of novel receptors. They are not natural receptors, they are synthetic receptors. We gave those the name of Chimeric antigen receptor. Of course, I will discuss these receptors very shortly. The kind of targets, the molecules that these receptors recognize are different from what the immune system normally recognizes. And some years ago, at Memorial Sloan Kettering Cancer Center, we thought that Cd19 would be a very good target for some cancers, leukemias and lymphomas, and I will discuss that. To bring this to the clinic, we had to address a number of new questions on how to engineer the human system using new cell manufacturing approaches. And this is what my colleague Isabelle Rivière, who's here today with us and meeting with some of you later today, has established at Memorial Sloan Kettering. And finally, to bring this to the clinic, we had to discuss this with the U.S. Food and Drug Administration, the FDA. The FDA had never seen therapies like this, and there was a very good dialogue between the academic center and the regulators in the U.S. to come to a consensus for protocols of manufacturing and clinical trials. All of this brought together in one program led to the initiation of the first Cd19 CAR therapies. So I would like to remind you how the immune system normally recognizes its targets, including cancer cells. To the left is the physiological, the normal receptor. This is the alpha-beta heterodimer that normally recognizes HLA peptide complexes that can be found on the surface of cancer cells. This alpha-beta heterodimer alone does not signal. It is associated with what is called the Cd3 complex, made up, as you see here, of multiple chains. Note this green one, this zeta chain, which I will come back to later. This complex directs the specificity of the T cells and initiates immune responses. But it is not sufficient to induce a productive, efficacious immune response. For that, you need to engage multiple other receptors, which are known as co-stimulatory receptors. Co-stimulatory receptors fall into two main families of molecules. Members of the immunoglobulin superfamily, such as Cd28, ICOS, and many others, and members of the TNF receptor family, such as 4-1BB, OX40, and many others. It is the concerted action of these multiple receptors that directs clones of T cells to produce efficient immune responses. When we started this work many years ago, it seemed too complex to genetically control so many molecules. So that created the need to design receptors that in a single molecule could accomplish multiple tasks. And to the right here is what we called a second generation chimeric antigen receptor. and it has 3 canonical components. On the extracellular portion, the SCFV, the single-chain fraction variable derived from an immunoglobulin, instructs the T cells what targets to recognize. When they bind to the cancer cell, this initiates T cell activation through this component, which is, you recognize here, the cytoplasmic domain of the CD3 zeta chain. This directs killing of tumor cells, but killing is not enough. The T cell has to expand and remain functional over several weeks to defeat the cancer. And so the third component, which we introduced at Sloan Kettering in these receptors, which defines what we call the second generation CAR, augments the functions and the persistence of these genetically engineered T cells. We had to identify a target, first for experiments in mice, but also a target that we thought we could bring to the clinic. And we identified at Sloan Kettering Cd19 as a possible candidate target. Cd19 is on the cell surface of cells, and therefore it can be recognized by a CAR in the T cell. Cd19 at the time was known to be involved in B cell activation, but found on the vast majority of leukemias and lymphomas. At the time, it was not a very popular molecule. It is now, today, a popular molecule. And we were interested because it was expressed at a fairly high level, hypothetically on tumor-initiating cells, and also in normal cells, which is not ideal, but only on normal B cells. And as you all know, physicians know how to manage patients who lack B cells, and so even if it is not ideal, it is manageable. Because we knew that the CAR T cell could not distinguish between a cancer cell and a normal cell. A CAR T cell will kill any cell that expresses CD19. And this is from the first report in the literature, which is already 15 years old, where we provided the first demonstration that we could take human T cells, engineer them with a CAR specific for CD19, and in these mice introduce B cell malignancies, lymphoma, Burkitt lymphoma, ALL. In this case, it's a Burkitt lymphoma, and all the clinicians here will recognize an FDG PET scan. And you see the cancer here, and the cancer is up here and in the spinal areas. And a single infusion of these CAR T cells could induce durable remission. If you give more T cells, all the mice can survive. It is a single infusion of human T cells treating and actually curing established lymphomas or leukemias in mice. We then decided that we should bring this to the clinic. Of course, there was no industry interested in these therapies at that time. So we had to establish the manufacturing inside our center. We had to build a so-called GMP facility that follows the rules of good manufacturing procedures. And this was accomplished by my colleague, Dr. Riviere, who's right here. She designed this with engineers and architects, had it approved by the Food and Drug Administration, and then developed this manufacturing process where we collect cells from patients by apheresis, activate the T cells. We use the only commercially available GMP grade material for that. These are iron beads coated with antibodies specific to CD3, CD28, because we want to purify the T cells from the apheresis. We want to expand the T cells to make the dose that we will infuse the patient. And in the process, we want to genetically modify them. And if the cells divide, the efficiency of that procedure is very much enhanced. We used viral vectors to introduce the gene coding for the CAR. We produced them in our center in Dr. Riviere's facility, and the T cells were cultured in these bags. And after 9 to 10 days, the beads were removed on a magnet, and then the cells can be frozen down or quickly tested for immediate release and infusion to the patient. This manufacturing at our center has a very high success rate, 97%. And those rare failures, the 3%, they are not technical failures. They are patients in whom the T cells, even if we try again, fail to expand. We reported the first results of this therapy in acute lymphoblastic leukemia, ALL, in 2013. And we reported on the first 5 patients we treated. These were adults with refractory, relapsed ALL. And here's an example of 2 of those 5 patients. This is the bone marrow, the day -4 or day -1 before CAR T cell infusion. And here's a FACS profile where you see CD19 here on the x-axis. Subjects 4 and 5, you see here, have a lot of leukemic blasts, 80% or 70% in these 2 individuals. After the CAR T cell infusion, you see here the complete elimination of any CD19 positive cell in subjects 4 or in subjects 5. As you see here, there are no more CD19 positive cells, but hematopoiesis is recovering. Neutrophils, platelets, red blood cells come back up. This is a targeted therapy. It's specific for CD19. This is normal hematopoiesis, except that it cannot produce B cells. These responses would qualify as complete responses. They are more than that. They are molecular responses. If you extract genomic DNA from bone marrow and do deep sequencing, I'll look again at subjects 4 and 5. In the marrow of day -4, day -1, all of these are malignant transcripts per fixed amount of genomic DNA. But you see here day 11, 1,000, day 24, 600, and sometime after day 24, it becomes undetectable. In subject 5, even more rapid. As early as 8 days after T-cell infusion, there are no longer any leukemic blasts detected by deep sequencing. This qualifies as a molecular response. As you may know, colleagues at the Children's Hospital of Philadelphia who read our papers and got interested in our approach did something similar in children and also found very good results. So we started with adults, and CHOP in Philadelphia proceeded with childhood acute lymphoblastic leukemia. And so later that year, we were very pleased when Science proclaimed, every year Science has the scientific breakthrough of the year. This could be on artificial intelligence or neurological disorders or mathematics. And that year, they proclaimed cancer immunotherapy to be the scientific breakthrough of the year. And this was for 2 forms of immunotherapy. One known as checkpoint blockade that uses antibodies to augment immune responses in patients who are lucky to already have some immune response. And the second was for CAR therapy. So other centers tested CD19 CAR therapies, and all of them reported these exceptional results. I say exceptional because it is very rare that in a phase 1 trial where you enroll patients that are refractory relapse with such a short life expectations that you would see responses on the order of 60, 70, 80, 90%. Usually a few responders in a phase 1 trial is sufficient to call it a success. And this was reported from 3 centers, Sloan Kettering, National Cancer Institute, and University of Pennsylvania. And so the scientific and medical community in the US immediately embraced and believed these results. And so did the press, and so did the patients. And you can see that in pediatric or adult ALL, these responses were very high. They're also high in non-Hodgkin lymphoma and chronic lymphocytic leukemia. Not quite as high, but still very remarkable. Again, all of these patients have refractory, chemo-refractory disease. Some centers use gamma retroviral vectors, some centers use lentiviral vectors. It didn't make a difference really. And there are two kinds of designs of CARs. Some use the Cd28 co-stimulatory molecule that I showed you in an earlier slide, and some use another co-stimulatory molecule called 41BB. But as you see, all of these gave very comparable results. And so last year, in 2017, it was a historic moment for us when the Food and Drug Administration, the FDA in the US, approved for the very first time 2 CAR therapies. One uses the CD28 co-stimulatory domain that I mentioned that we described and developed at Sloan Kettering, and the other one, a 41BB co-stimulatory signal. Both of them are specific for CD19. And so one is now sold by Novartis for the treatment of pediatric ALL and some forms of non-Hodgkin lymphoma. And this one, called Yescarta, is now sold by Gilead for some forms of non-Hodgkin lymphoma. The importance of this for us was that this was the first time that a cell therapy using a genetically engineered cell was approved as a drug, the very first of its kind. Here's a summary of our results in acute lymphoblastic leukemia. Again, our studies being in adults. And we recently reported in the New England Journal of Medicine on the first 53 patients that we treated. And the overall complete remission rate is 83%, confirming the earlier results in small series. And if we look at patients who have a low disease burden, less than 5% blasts or more, there's no difference in the rate of induction of a complete response. Some of these were enrolled on this protocol having relapsed from a bone marrow transplant, 19 of them, others had not received a transplant. This did not make a difference. Some of them got 2, 3, 4 or more prior chemotherapies. No statistical difference. Some of these patients have Philadelphia chromosome P53 mutations, other anomalies. It does not make a difference. We have to give 1 round of chemotherapy called the conditioning chemotherapy before infusion of the T cells. Our experience is more limited with fludarabine, but in 2 different ways that does not make a statistical difference. And finally, age, which in adults is a poor prognostic factor. You could see that we have fewer patients that are older than 60, up to about 78 years of age, but this did not make a statistical difference. What I want to highlight is how robust this procedure is. A patient may have received 2, 3, 4 chemotherapies, maybe a transplant, may have Philadelphia chromosome, maybe age 60. The probability of inducing a complete remission remains high. And this shows you that the process of collecting patient cells and engineering them with the CD19 CAR is really robust. Now, a complete remission does not mean a cure. And for that, of course, we have to wait much longer. And so this therapy, as I will show you, saves lives, but it still can be improved. The 15% of patients who did not go into molecular response will soon progress from their disease and die from their disease. They still live a bit longer for some of them than they might have, but it's clearly not curative. But even if you do go into a molecular remission, not everybody will survive. The good news is there's this long tail here of true survivors, and I think we can now say that the first patients that we treated 6 years ago, 5 years ago, are cured. But many will relapse, as you see here, a bit more than half. In children, perhaps because their T cells are of better quality, the survival is higher, although there too, close to half of them may eventually relapse. Two observations that we made. You remember the high tumor burden and low tumor burden. It's the patients with high disease burden that relapse the most. Those with a lower tumor burden do better. And that tells us that we should give this treatment earlier when the tumor burden is lower for a better result. The second point is that in some of these patients, we propose a transplant because, as you know, if you succeed to induce a second remission with the CAR therapy, for example, or through other means of chemotherapy, you will typically consolidate that response with a bone marrow transplant in ALL. So we proposed initially a transplant. Some patients got a transplant, but many did not. They refused the transplant, they were not fit, there was a donor issue. And interestingly, about half of them received a transplant and half did not. And as you see, it did not make a difference. So this does not prove that a transplant is now unnecessary. For that, we would need a prospective study, but it is at least suggestive that the CAR therapy may indeed be curative on its own. A word about the toxicities. There are three main toxicities, one that I already mentioned. The CAR T cell cannot distinguish today between a leukemic cell and a normal B cell. So it could cause a B cell aplasia, and I showed you some of that before. It turns out it is not as big a problem as we thought it might be. First of all, physicians know how to manage B cell aplasia with IVIG. But also, CAR T cells do not persist in most patients for very long periods of time. And when the CAR T cells disappear or become dysfunctional in the patient, then B cell lymphopoiesis recovers. And so this is no longer a problem. It is a problem, however, in a minority of patients where the CAR T cells persist indefinitely, in which case the patient could be in B cell aplasia for a very long time, and that will require modifications of the CAR T cells in the future. But there are two other toxicities that had not been anticipated in the preclinical studies in mice, which were found in the clinic. And the first is one that we call cytokine release syndrome, or CRS. Most patients developed a fever soon after getting CAR T cells, which accompanies, any strong immune response will give you some fever. But in some patients, this fever is elevated and persists for days, and may also be accompanied by hypertension, cardiovascular complications, and respiratory problems, and these patients have to be transferred to an intensive care unit, ICU, for management. This is reversible. There were deaths in the very beginning at our center and at the University of Pennsylvania National Cancer Institute because nobody had anticipated this, and these were very surprising responses. Surprising, although in the end very logical. They are the consequence of the normal function of a very intense immune response. It is because the T cells are doing their work that these cytokines responses are induced. So there are ways to manage this response today, and I will tell you more about that later. The other toxicity are neurological changes. In our first paper in 2014, we classified those as part of the CRS, but now we and others agree that this is a separate complication. It often occurs with the CRS, but sometimes it occurs on its own. And some patients have, again, it is transient, like a CRS. Global encephalopathies, the patients are lost. Very spectacular, but transient aphasia, and sometimes seizures. I won't say more about this today because this part is really poorly understood. For those of you who are familiar with blinatumomab, the Cd19 engager, this same two toxicities occurs with blinatumomab. So it's not only CAR T cells, but certainly those strong immune responses to CD19 can induce these effects. So in this case, if you remember the subcategories of these 53 patients, there is one variable that really stands out as difference in whether the patient will develop CRS or not, and it's the tumor burden. Patients with low tumor burden are very unlikely to develop CRS. Those patients get CAR T cells and they go home. The patients with high tumor burden, more than 5% blasts here, those are the ones who are at risk of developing the CRS, and those are the patients that we keep in the hospital. So this is by introduction, a rapid overview of Cd19 Car Therapy. And now I would like to discuss some future directions, but I understand that some of my slides may be a little bit scientific, so I will, if you don't mind, not focus on every detail, but just give you some insights into what we are doing now to further improve this CAR therapy. And so this T cell has to work, kill tumor, and if there is tumor left, it has to multiply and continue its work until the tumor is gone. There is a process, however, that can adversely affect the function of lymphocytes, well known in cancer patients and in chronic viral infections, which is the exhaustion of T cells. The T cells don't die, they persist in the body, but they gradually lose their function. And this is called exhaustion. And we have found that in mouse models and in patients, sometimes the T cells will exhaust, and therefore they are no longer therapeutic. So we are very interested in understanding the exhaustion and finding ways to make T cells that will not exhaust and persist in a functional state longer. And we have now come up with a genetic approach and a signaling approach to reduce the exhaustion into CAR T cells. And then I will discuss what we have done recently to better understand the cytokine release syndrome, and after that I'll conclude. I will go through these three studies in a accelerated version, if you don't mind. This field began, the field of T cell engineering, I think began at MIT. Certainly, I was not aware of other people doing this many years ago, back in 1989. And in 1992, we showed that we could indeed introduce genes into T cells. They were mouse T cells, not yet human. And for that, we used retroviral vectors. This is what we use in our protocols at Sloan Kettering. This is what is used today in the Gilead product that is approved for non-Hodgkin lymphoma. In subsequent years, investigators use other genetic tools to introduce the CAR cDNA into T cells. Some used lentiviral vectors, and some use what's called transposons, DNA molecules, such as Sleeping Beauty or piggyback transposons. All of these work. They're all efficient to introduce genes into T cells today. They share one common feature, and that is that when they introduce the DNA in the cell, the DNA has to go on a chromosome to remain part of the T cell. The site of integration is random. There are millions of potential integration sites. And for those of you who studied genetics, you will know that a randomly integrated transgene is subject to chromosomal position effects because the chromatin surrounding the transgene will modify how the transgene works. This is also known as gene variegation. Over the last decade, there has been tremendous progress in the field of gene editing, providing new tools to target specific regions in the human genome. And from meganucleases many years ago to now CRISPR-Cas9, these tools allow to induce a selected break on a chromosome, and at that position where the DNA is broken, you can now insert the CAR cDNA. So we have been for several years following this new technology, and we used it to ask, what difference does it make if we can select the integration site of the CAR rather than rely on random integration as we have done till now? And we studied a few positions in the human genome, but one of them is very interesting for us. And it is the location that codes for one of those T cell receptor chains that I showed you in the T cell receptor in the beginning, the alpha chain. You can do the same with the beta chain. We introduce the cDNA coding for the CAR exactly in this locus. And we do it without using a promoter. When this cDNA is inserted in the patient's, or human anyway, T cell genome, it is controlled by the promoter enhancers and other genetic elements that normally control expression of the T cell receptor alpha chain. And here's a typical experiment. Human peripheral blood T cells electroporate the Cas9 nuclease, the guide RNA that tells it where to cut, and it cuts right here. And then the donor DNA, which is this one here, which is carried by what's called an AAV6 vector. So here's a control first. If you just expose the donor DNA, all of these T cells do not express the CAR as expected, and they all express their T cell receptor. If we now introduce the Cas9 and the guide RNA, a lot of these cells now lose the T cell receptor on the cell surface. Why? Because we cut the T cell receptor alpha locus. And now if we start adding more and more of the donor DNA, we get these cells. These cells are very interesting to us. They are even more synthetic T cells now. They are still T lymphocytes, but they have no more T cell receptor, the normal receptor of T cells, but they have the CAR, and only the CAR can tell the T cell where to go and what to do. The surprise in this study was when we functionally evaluated these cells. And we compared these gene-edited cells that we call TRAC CAR T cells. TRAC because it's the name of the T cell receptor locus in genetics. And when we compare them to the cells that we make today, so the cells that we make today will cure patients, as I showed you before, but in these experiments, we infuse human T cells in mice that have a human leukemia at very low T cell dose, very low dose. This is a low dose for a mouse, 200,000 or 50,000 T cells. And you see that these cells don't work anymore. These are days, 100 days, and all these leukemias progress. Of course, if we give 1 million cells, we can cure these mice, but not at this dose. However, the TRAC-edited cells can still induce tumor delays at this dose and cure some mice even at this low dose. So we know that they are functionally superior. We know that this superiority does not come from having disrupted the T cell receptor alpha locus, because here's an important control. In these cells, we disrupt the T cell receptor and then introduce the CAR at a random location. And you see these cells are no better. So what it means is that these T cells are superior in their function not because we remove their T cell receptor, but because the CAR is now expressed from that specific locus and promoter. I'll be quick on the biology. I apologize for the scientists in the room. I think more of you are clinicians. But we tried to understand why putting the cDNA at that position makes a better T cell. It turns out that when the CAR T cell is exposed to the antigen, Cd19, the CAR here you see disappears, or at least is vastly reduced on the surface of the T cell at 12 hours, and then it comes back. at 36, 48 hours, it comes back. So it binds to the tumor, kills the tumor, the CAR goes inside the cell, and later it comes back. We know from experiments that I won't show that it is degraded. It is degraded so the T cell has to make new receptor to replace it. When the CAR is expressed from the lentivirus or retrovirus, different things happen on the cell surface. It is not completely internalized, and in fact there's more expression soon after encountering tumor antigen. And this is summarized in the following slide, where from the TRAC locus, it disappears from the cell surface and then comes back. When we engineer the cells with the viral vector, the reduction is less. This is an average of 12 experiments, and it comes back fast, higher than the initial result. These cells that express lots of CAR are ready to kill again. They can kill very fast. However, what we conclude from these experiments is that these cells that lose the CAR can rest a little bit. They have a little break, and then they are ready to kill again. And the T cells in the patients have to work not for 18 hours or 36 hours or 120 hours. They have to work for weeks. And if the CAR comes back to the surface too soon, they eventually go into exhaustion and they become dysfunctional. So we are now preparing to test with Dr. Riviere the CRISPR-Cas9 T cell in patients, and I'm hoping that in a year I have data to report to you on this. We've tested many locations in the genome, I will go through this, and we find that most sites are not adequate. The TRAC locus is very good, and we are continuing to explore parts of the human genome where it would be most effective to express the CAR or other molecules. And I think this will be very fruitful to design better immunotherapies in the future. Now, the second approach I want to discuss to reduce exhaustion are changes in the signaling of the CARs. So this is our first CAR, the one that's now approved for lymphomas, which has the CD28 zeta, and the one that was used at the University of Pennsylvania, which came from St. Jude Hospital. It was described by Dario Campana, Imai et al., at St. Jude, and then developed at Penn, and today's Novartis product are shown here. But researchers, more and more researchers, including us now, are designing new CARs and testing better features for a better immunotherapy. And not only CARs, there are chimeric cytokine receptors, chimeric co-stimulatory receptors. I just show this to make the point that these synthetic biology approaches have enormous potential to design new kinds of immune responses in patients. So I would like to show you the next design that we want to bring to the clinic. And we start from CD3 and CD28. These are the CAR T cells that are in the clinic today. This is, if you like, the Sloan Kettering molecule or Gilead molecule or NCI in Washington molecule. This one that uses 4-1BB was used at University of Pennsylvania and Seattle, and now it's Novartis. And this is what's called a first generation CAR, which does not have a costimulatory domain, red or green, just activation. And when we put T cells in mice where the CAR only gives activation, there's no therapeutic effect. These CARs don't work. Activation without co-stimulation is insufficient. And here's the effect of this 28Z and 4-1BB CAR T cells. Again, by intention here, low dose, which is not sufficient to cure the mice. And there's not much long-term survival benefit. But in the mice, we analyze what happens to the CAR T cells. In patients, we try to do that. But there's only a limited number of samples that we can take from the patients. Of course, in the mouse, we can study human T cells and make measurements in many tissues at many time points. And here's what we find in the mouse model of acute lymphoblastic leukemia, ALL. So we're now looking what happened. These mice had the ALL, CD19 positive, we infuse the T cells, and we see what happens 7, 14, or 3 weeks after the CAR T cell infusion in the bone marrow, which is the site of the leukemia. So the first generation CAR T cell has a small amount of T cells, and this 28 and 41BB CAR T cell is much more abundant. This is a logarithmic scale. There are 30 or 40 times more T cells. So this shows you how the co-stimulatory domain inside the CAR helps the expansion and persistence of T cells. But while their cell number is the same, their effect on the tumor is very different. The CD28 CAR has killed the tumor already, but not the 41BB. The 41BB is not as aggressive a killer cell. However, the CD28 CAR T cell, you see, diminishes in numbers. You see 10 to the 5 here. This is a few 10 to the 4 and lower by day 21. And this is in contrast to the 41BB CAR. In fact, at day 14, there's even more than on day 7. And eventually it will go down in number. So while these 41BB CAR T cells are less active, they expand more and persist longer, and also succeed in this model in eradicating the tumor. The reason I show you these details is that, first of all, they are very reproducible from one experiment to another, and secondly, because they are very similar to human patient data. It's difficult to analyze human patient data. There are fewer data points and different trials, different B-cell malignancies, different T-cell doses, different conditioning regimens. It's not so easy to compare. Of course, we all try to compare and to learn more. In the mouse model, you can reduce the variability, and these findings are very similar to what we see in patients, meaning that the CD28 CAR is very active but disappears in a few weeks after infusions. Whereas the 41BB CAR, nobody could know really that it was less active. You needed the mouse model, but they persist longer before going away. As you recall from the clinical summary, both CAR designs have shown very similar results in ALL, NHL, and CLL. However, with different kinetics. What we learn from these analyses is that there are different ways to now improve the CARs. The CD28 CAR is very active, but doesn't persist. Can we design it so that it persists longer? Another approach would be to take the 41BB CAR, which persists, but is not so active. Could we keep the persistence and increase its functional capability? So we pursued the first approach, and we now have introduced various mutations in the CD28 CARs. And here we focused, this is not published yet, but we focused on the activation part from the CD3 zeta chain, the green component that I showed you in the beginning. And so we have different mutations in what are the activation domains in this molecules. They're called ITAMs. There are 3 of them, 1, 2, 3. And here we delete or mutate these 2, keep 1, here we keep this one, here we keep this one, 1XX, X2X, XX3. And this is the same mouse model testing human cells. This is the CAR that's in the clinic today in patients. And you can see now when we use this 1XX, the function is far improved over this one. And that's what we see. I will go quickly through the next T cells, through the next slides, which show you that the 1XX is slightly more abundant than the wild type. It is less exhausted, which to us explains why these mutations induce superior results. And so what we now realize is that we can modulate, direct the differentiation of CAR T cells under our control by dosing the strength of activation in the CAR. The wild type gives you great activation, but limited persistence. And this one, which is the one we will bring to the clinic now in 6 months or so, has the right combination of enough activation of effector functions, but without repressing the memory program, which these cells do. They repress the genetic program for memory, which is why the cells are short-lived. And when we use the XX3 model, I didn't show you that, I'll just summarize. Now we could get great memory. However, those had very poor function. So this is what we are now bringing to the clinic. I want to now discuss, before making a few general conclusions, progress we have made in understanding the toxicity. You remember the CRS, the cytokine release syndrome. When the FDA approved the first CARs last year, it was approved in a very particular way. The CAR therapy could only be administered in centers in the United States where the personnel, meaning the physicians, the fellows, and the nurses, were specifically trained by the companies to manage the CRS. Of course, at some academic centers like ours, we didn't need the training because we did that before those companies. But this is a restriction. Only certain centers can administer the CAR therapy. And it's also a restriction in a general sense, is that, we would love to be able to abolish this toxicity, and then you could give more CAR T cells for more diseases at many more hospitals. But to do that, you have to understand it. And this was not so clear from patient studies. So this slide is just to show, it's a very updated slide, that there's now a classification of CRS, grades 1, 2, 3, 4, 5. Severe is grades 3 and 4, 5 of course. And you can see that in different trials, from ours at Sloan Kettering, or these commercial trials, or the NCR trials or CHOP trials, everybody sees CRS. Mostly in ALL. You see here, whether it is in adults or in pediatrics, in ALL. Almost half of patients. Again, we know those, as I showed you, are the patients with high tumor burden. There is less CRS in lymphoma, nobody knows why, but it's still an issue. So there is a way to manage CRS today, and it's the group at CHOP that empirically found that you could give an antibody to IL-6 receptor, and in some of the patients that's enough to control the CRS. But not always. And we have algorithms for treatment. This is what we use at Sloan Kettering. If the patient doesn't quickly improve or worsens very soon after, we then proceed very quickly to high-dose lymphotoxic doses of corticosteroids, and those are usually effective. Physicians were initially reluctant to give those because the concern is to limit the therapy, because you don't want to kill off the T cells. It takes time to understand the consequences of these interventions. So far, it doesn't seem like it compromises the therapy, but nobody is entirely sure yet. We need to have, thousands of patients treated before we have a clearer understanding of this. But this treatment is empirical. It's clinicians who very smartly found ways to control the CRS. What you really want is a rational understanding of CRS. And for that, you need a model. And we tried for years to get a model. Others tried too. And some said that you could not model this, that it's only a human circumstances. But we now succeeded in creating a mouse model. And it is different from the therapeutic models that I showed you before. It is still human T cells in an immunodeficient mouse, but it's a different strain of mice. These are skid beige mice. And we have to establish very large tumor burdens. And the bone marrow is too small, so we have to go in the peritoneal cavity, and we have to wait for 3 weeks. And then we give CAR T cells, very high doses, not the low dose that I showed you before now. This is a very high dose. And now the mice, as you see, within 2-3 days, they lose weight. They don't have fever like our patients, but they have hypothermia, but that's normal for the mouse. And some of them start dying. And also in patients, we see C-reactive protein going up very soon. In the mouse, it's SAA3. This goes up in the mice. So this looks like CRS in patients. we analyzed 3 papers, our own data, and the data from 2 other centers, created a list of cytokines that all centers agree are elevated, or some are not elevated. Some are elevated in patients. So this is a consensus list. And in our mouse model, you can see remarkable similarity. What is elevated in patients is elevated in the mouse model. What is not elevated in patients is not elevated in the mouse model. So we think the mouse model has some similarity to the human condition. Now, these are human cells introduced in a mouse. So that creates a very interesting situation. When the cytokine is high in serum, we can tell if it comes from the mouse or from human. So GM-CSF, IL-3, interferon gamma, and many others are high in the serum and human. They come from the T cell. But the IL-6, you remember, which is treated today with tocilizumab, the blockade of the IL-6 receptor, it is not human. It's from the mouse. So it's not the CAR T cell that's making the IL-6. It is cells in the recipient, presumably myeloid cells. And one further test that we did to show that this again has similarity to the human is we could now prevent mortality by blocking in the mouse IL-6 receptor. So this is not tocilizumab, this is an antibody that blocks the mouse IL-6 receptor, and we could prevent mortality. So what are the cells that are producing IL-6? So I'll go quickly. These tumors in the mouse have lots of these brown cells are myeloid cells. They're mouse myeloid cells in the human cancer here. And we find lots of myeloid cells, granulocytes, dendritic cells, macrophage, monocytes. And so we ask, which cells are making this interleukin-6? It turns out that all of them do if they are in the vicinity of the tumor. So they turn on, this is a fold induction of IL-6 in highly purified myeloid cells, dendritic cells, macrophage, monocytes, and they all turn on IL-6. What I'm not showing you here because of time is if we look at monocytes, macrophage, and other tissues, they are not elevated in the lung and the bone marrow. So it's not a systemic disorder. It's not all over the body. It's the myeloid cells that are close to the tumor that induce the IL-6. And so the tumor is the model is not anymore what we thought it was, which is CRS is only caused by CAR T cells seeing the tumor and secreting cytokines. No. CAR T cells see the tumors and also interact with other myeloid cells close to the tumor, and that causes the toxicity known as CRS. So I am going to move on to, there are other ways now of treating the CRS, and we are now going to open trials later this year testing other ways of preventing the CRS, not through IL-6 receptor blockade, but by blocking, in fact, IL-1. But I should accelerate to finish. I'll just say this, that now what we realize is that the CAR T cell, yes, attacks the tumor with the CAR, but it can also activate myeloid cells, such as the macrophage, that are surrounding the tumor. And this is very interesting. First, we see here that it contributes very much to the toxicity, but we also realize now that the CAR T cells can be doing all kinds of things to the myeloid cells, including maybe helping the tumor rejection, and we intend to study this further. So, I have a few slides to summarize. For many years, T cell therapies focused on isolating good T cells from patients or from a donor. And the clinicians in the room will know these lymphokine-activated killer cells, LAC cells, in the late 1980s. Then also in the early 90s, tumor-infiltrating lymphocytes, T cells. Of course, in bone marrow transplant, the donor leukocyte infusion. And at some point, the isolation of virus-specific T cells for EBV, CMV, or influenza. And all of these, though, are what I call natural adoptive cell therapies. You look in the patient or in a donor for good T cells that are therapeutic with limited toxicities. However, the path that we propose is now to not look for those natural cells, but to engineer and produce those cells, at least for patients who cannot make them on their own, and maybe for all patients. And so this is based on gene transfer and soon also gene editing and all kinds of synthetic biology that I tried to quickly illustrate today. And the paradigm for this is Cd19 Car Therapy. Where should we apply this? Well, I want to put this in the context of all immunotherapies. As you know, checkpoint blockade, antibodies blocking CTLA4 and PD-1 or PD-L1 have been very successful in many cancers. Interestingly, this success is linked to the mutation burden. This is a famous slide, modified by me here, but that shows you the different mutation burden in different cancers. The very high, for smokers, I see many smokers in Russia, by the way, I'm a little worried. Lung cancer, of course, you can induce all the mutations you want every time you smoke, and you can successfully induce hundreds and sometimes thousands of mutation in your cancer. Same thing with ultraviolet and myeloma, and melanoma, And of course there are other cancers with high mutation burdens. And this is where checkpoint blockade works the best. But our success now in ALL, or in particular, is on the left side here, a very low mutation burden cancer. AML, a diagnosis, typically has 13 mutations, not 500 or 2,000 mutations. It is far less likely to be immunogenic in the patient, and therefore it is very unlikely that checkpoint blockade will work. And so we think that CAR therapy is especially relevant in patients with low mutation burden where, again, checkpoint blockade may not be so effective. And of course, we don't exclude these cancers because even in these melanoma and lung cancer, there are patients with low mutation burden. This was written last year and it has already changed. This was in December of last year. There were about 20 CAR targets in clinical trials around the world. In the US, over 40 biotech companies and big pharmaceutical programs have now started CAR therapy programs. There were over 80 CAR targets, it's over 100 already today, that were reported in the literature. I don't think that most of those targets will be successful in the clinic because they may be imperfect, but if even a fraction succeeds, this will be a phenomenal change. And in December, there were 240 trials listed on clinicaltrials.gov, which is a place where you can find all these protocols listed. And already today, it has exceeded 350, I'm told, and this is only 6 months later. The two countries that conduct most of these trials are the United States and China. China has now gone very far in CAR therapy. Because they have a long tradition of cell therapies in their hospitals, and they added the genetic engineering into it. And of course, we're very excited now to see Europe, and especially Russia, now starting these trials, and especially at this center here, where I am so excited to see the first results of Mikhail Meschan, who told me about the first patients that he has treated here. It's an incredible tour de force that he has been able to do that on his own with your help, I guess, at this center. And again, his first results are as good as ours. So his first efforts are truly impressive. The last point I want to make, and some people still think that cell therapies are a challenge to develop in our medical systems, whether it's the United States or Russia. Indeed, it's a new form of medicine. You have to process cells. The pharmaceutical industry, for the longest time, was refractory. They know how to make chemicals, they know how to make proteins, antibodies, vaccines, clotting factors, but not cells. But what the Cd19 Car Therapy has done is convinced the Novartis, the GSK, Pfizer's, and others to now make cells as medicines. And there are truly more and more centers now contributing to this effort. So we think that the meaning of Cd19 Car Therapy is, of course, first for patients with refractory leukemia and lymphoma. I hope this will become a first-line therapy, and there will be efforts for that pretty soon. And of course, we hope that this will pave the way for many more cell therapies. There are today, this is the tip of the iceberg, promising results in multiple myeloma, reported from several centers in the US and China. AML is a very big challenge. Let's see what happens, but there are certainly several centers, including ours now, that are getting ready to tackle AML. The big unknown is how successful this will be in solid tumors. The first trials, which we did not do, were not so successful. We don't think that the CARs that currently exist are best suited for solid tumors, but hopefully this will come. Now, T cells are not just relevant for cancer. They're relevant for autoimmunity. And there was a report last year in Science showing a treatment of a form of pemphigus, again, human T cells in a mouse model, effectively eradicating T cells that cause pemphigus. We are also starting at our center, and I think a few others, to design T cells for hard-to-treat infections, such as fungal infections. We are interested in that. CAR T cells can be engineered for severe infections as well, and maybe chronic infections as well. In organ transplantation, solid organ transplantation, the engineering of regulatory cells, Tregs, has begun. And of course, behind all of this is this big, big field of regenerative medicine using all kinds of stem cells, from bone marrow, cord blood, to induced pluripotent stem cells. And we think that now, having convinced some in the industry and some hospital administrators, and in the US, quite significant investment, that there's a great future for cell therapy. And to me, that justifies any big center, big medical center, should now have, we think, its own cell therapy facility. And I say that because we believe that academic centers will play a critical role to develop this. Companies, it's great to have companies. They can make lots of this. But there are so many diseases, so many CARs, so many cell types to study, sometimes rarer diseases, that companies will not want to pursue. But this field will continue to be driven by academic centers for the decades to come. My slides had pictures of fellows that contributed to some of the work that I mentioned. My two very long-term collaborators are Rainier Brentians, who was a fellow of mine, who did the first mouse experiments in Cd19, and now has established a service of cell therapy at my center. And really, Dr. Riviere, who again is here with me today, who makes all of this possible, because without her team and without a GMP facility, you can publish lots of mouse papers, but you can't treat anybody. And so she's really the one that made all of our program become a reality. And I thank you here for your attention. Thanks. What Mikhail is referring to here is to generating cells that are not autologous. The current therapies are autologous, which imposes manufacturing for every patient. There's a reason why they are autologous. If you take my T cells and engineer them with a CAR and give them back to me, these T cells can function in their original immunological context. if we take one of your T cells and put a CAR in your T cells and infuse them to me, the CAR will tell your T cells to attack my leukemia. But, as you know, your T cells will attack me, if you like, and trigger a graft versus host disease. But I will also attempt to reject your T cells. So there are two problems, very classic problems of bone marrow transplant biology, really. Graft versus host disease and host rejection. So that precludes right now the use of allogeneic T cells. But there are several groups, academic and also now industrial, who are thinking of ways to modify allogeneic cells. This would reduce the needs for manufacturing. And so there are a few approaches that people have used. One is to use virus-specific T cells that retain their own receptor, the idea being that they will not cause graft versus host disease. And so they would have a CAR, but you would engineer virus-specific T cells. The other approach, which I alluded to somewhat, is with gene editing, you can destroy the T cell receptor. That's even safer than this, because this, although may not often cause graft versus host disease, there still is a T cell receptor. Is deletion of the T cell receptor and creating these cells that I showed you that are TCR less negative, but CAR positive. Another approach, less popular but reported in the literature, makes use of lymphoid progenitors that you can make in the laboratory from cord blood and adult marrow that engraft in the thymus, acquire tolerance to the host, and therefore do not cause graft versus host disease. A common feature to all of these are interesting approaches, and there are a few trials that have begun testing both of these, and there's not enough data to see how effective they are. However, I wish to point out that they only take care of one problem, which is the T cell attacking the host, the graft versus host disease. They do not take care of the other problem, is me now rejecting your cells. So these protocols are only implemented in severely immunocompromised patients, which is fine or acceptable in a post-transplant setting. But should you develop these CAR therapies for patients, with other cancers, with glioblastomas or breast cancer or whatever, we don't believe that immunocompromising the recipient is the future of this therapy. So we have to design smarter T cells that can really avoid the immune response. And here too, there's a number of strategies that are in the public domain. Eliminating HLA expression from these cells is something that several groups are doing. Whether that will be enough or not is unknown. We think it will help, and we doubt that it will be enough. So we certainly are establishing different ways to control the immunogenicity of the T cells. But in all fairness, nobody knows how this will work. And this would, of course, be another major advance. You would still need manufacturing, presumably, in the academic setting, although at least it won't be one-to-one, autologous. It may be one donor for multiple recipients, and that would be a great advance.
Thank you. I really enjoyed your lecture. I have a few questions. First of all, you know that gene modification may be tricky. Either it would be lentiviral transduction or retroviral or transfection. So how do you ensure the sufficient percentage of transduced cells in your population? And how do you manufacture the sufficient amount of modified cells to cure the patients? And it raises a question about the overall cost of this therapy, because generation of lentivirus supernatant, for example, is very expensive. So,
yes. So 2 questions. One on efficacy. Well, that's what laboratories will work very hard to optimize. That's certainly what Dr. Rivera's group did over several years at Sloan Kettering, you can achieve today quite high genetic modification in patient cells. It is variable, though. And this is an unavoidable feature of autologous therapies. Every patient is a little bit different. And so there are some where the gene transfer is less successful. But again, in our program, and as I said, we treated over 250 patients now, we've never had a failed production because of insufficient gene transfer. Sometimes it is low. The dose is not a dose of total T cells, it's a dose of modified cells. So if the percentage is lower, then the culture has to be longer to expand more cells. And we know that a longer expansion is not preferred, but at least that's how it's dealt with today. Overall, though, gene transfer with high titer vectors is pretty successful, at least with gamma retroviruses and lentiviruses. In the case of gamma retroviral vectors, which have been around for the longest, they were the first vectors really used for human cell engineering. There are now extremely good packaging cell lines where you really can achieve high gene transfer efficiency. And Dr. Aguirre's group developed a little bioreactor just like this, where they can make vector for 400 patients in 10 days. And so that is very advanced. With lentivirus, as you say, it's more expensive and more challenging, but a lot of investigators have chosen to use lentiviral vectors. Transposons, until now, I said that they worked before, but they're not very efficient, and that imposes long cultures, and that has been very detrimental for those trials. Long cultures are not good. They impair the quality of the T cells. However, there are groups that recently have improved the transposons, and maybe those will become more useful, and those are a bit less expensive because they are non-viral. With regards to gene editing, I showed you high efficiency. It's 50, 60, 70%. And that's not just in deletion of a gene, that's in insertion of a gene. And that is amply sufficient to initiate clinical trials. So there are several approaches, and I think most of them have now reached a point of high reproducibility. But then that brings us to your second question, a very good question and very painful question, the cost. And what I mean by painful is that I think researchers like myself wanted to develop curative therapies, which a powerful immune response can do. And this is what inspires our effort and justifies the more complex logistics of dealing with cells rather than just pills. I mean, pills are great, but a lot of people do say that their goal is to transform cancer into a chronic disease. And I agree that that would be wonderful to have patients who live longer with cancer as a chronic disease. We're not there yet, but that's what the industry and a lot of academic centers say. We are looking for something different. We're looking for a curative therapy. And that is what justifies, in our mind, the development of a more complex therapy, if it can be administered once to be curative, and if it's affordable. So we don't control the price. It's companies and governments that control the price. And in the US, Novartis and Gilead negotiated with the branch of the US government and came up with the price of $500,000 for one infusion. We as academics are very saddened by this because we think it gives the wrong impression of cell therapies, that they are always going to be so expensive. To make the cells, and with all the testing today, costs about $40,000, $45,000. not $500,000, okay? So there's a big profit margin that the companies are building into their price. Now, I understand that companies have to pay, it's for profit, but it's also to pay the development of this new technology. They have to build facilities that did not exist before. And that is a really heroic effort. But still, we are saddened that they decided to charge such a high price. Again, it is not our vision. But this, interestingly, is now changing our research program. Because in my program now, we realize that we should do research not only to, find new targets, overcome the tumor microenvironment, prevent exhaustion of T cells, but we should now do research to decrease the cost. And we are doing that. That is, these allogeneic approaches, and also creating better T cells. The reason for creating better T cells is if they are superior, you need fewer. If they're better, you need fewer. And if you need fewer, the duration of culture goes down, the cost of reagents go down. Another benefit of having better T cells is that based on what we know, we predict the toxicities will be reduced. So making a better T cell should also, at least in principle, contribute to decreasing the cost. So that's what we can do to control the costs. After that, it's a matter of negotiation between companies and governments, and I guess different countries will come up with, will approve different prices.
Thank you for a wonderful lecture. I have two related questions. Yes. One, you said that most of the failures of CAR T therapy is due to their inability to multiply. So is there a way to predict in which patient it's going to happen so you don't even start? And another question concerns the non-Hodgkin lymphomas. Is the failures there also due to inability of the CAR T cells to penetrate inside and the access to the tumor? And can you also prevent looking at the microenvironment when it's going to happen, in which patient?
Yes. They're excellent questions as well. I may not have the full answer that I think you would hope for. The first question is about screening patients for the proliferative ability of the T cells. So we have not done that at Memorial, and yet our success rate of manufacturing is 97%. Now at CHOP, Children's Hospital of Philadelphia, nobody knew in the beginning that they were screening patients. I think they forgot to tell everybody. And we found out because many patients came to our center because they were rejected from CHOP. So we don't understand what's the test that they were doing. And they have an overall failure rate of 20%. So you would have to ask them exactly what their screen is and what they're doing. But as I said, our series had many patients that were rejected from CHOP, and we still have this high manufacturing success rate. So I guess that there's something that we're doing that overall allows for better expansion in vitro compared to them. Now, in the patients, however, we see that patients in whom the T cells fail to proliferate will fail the therapy. There's no doubt that this living therapy requires that the T cells go in the body and expand, and if they don't do that, the therapy will fail. There's a recent paper from Novartis Penn in chronic lymphocytic leukemia, CLL, that was just published 2 months ago, and that further attributed the success or failure to the content in a subset of memory stem cell-like T cells. Now, CLL is, I know we're in a pediatric hospital, and CLL is, a disease of older patients, more males than females, and often patients who have received multiple chemotherapies, as well as patients with a cancer, a CLL, that happens to be very immunosuppressive. So a lot of those patients have deficient T cells. That's one of the reasons people believe why the therapy is less effective. The quality of T cells in CLL patients is less good than in lymphoma or leukemia patients. But the biology of that, other than a reduction in the pool size of memory cells, is still not fully understood. Which brings us to the second question, I guess, which is, why is there this hierarchy between the three diseases? ALL, best response, NHL, quite good, and CLL, less good. Still not bad. You're still saving lives, by the way. And again, nobody really knows. There are two main possibilities. You alluded to one, right? Either, in the case of lymphoma, the T cells fail to reach the tumor, which would be a homing and trafficking problem, or they succeed in going to the tumor, but then the tumor microenvironment, different in lymphoma than, of course, leukemia, prevents these T cells from acting. So the data is limited. You know, you do fine needle aspirates from patients, and what we see in very few patients and few aspirates is that we can detect T cells by PCR, but we can't quantify that too well, but it appears that T cells get there, which would suggest that it's more the tumor microenvironment. However, there maybe is a limitation in trafficking. There's one interesting observation that we and our colleagues at Penn have in patients with CLL, which is maybe informative to a limited degree. And it's the following. In patients with CLL, they often have disease in their bone marrow and in lymph nodes and spleen. What we see is, in those patients, as I said, there's fewer complete remissions, and most of them are partial remissions. However, in those partial remissions, the bone marrow is clean. It's actually a complete remission in bone marrow. It's in the lymph nodes that there are issues. So I think that one of the reasons that this therapy is so successful in maybe the more aggressive disease in ALL is that cells traffic very well to the bone marrow, and perhaps the tumor microenvironment, as inhibitory as it might be, is not as inhibitory as it might be in lymph nodes. And I think that's all we know really right now. But it's important to find out, because if you understand where the problem is, then you can design the next CAR T cell to do better. Yes.
Thank you very much for your wonderful lecture, and my question is pretty much related to the previous one. It's again about those 3% of people that can be cured by CAR therapy. And I want to ask if, does this mean that CAR therapy is 100% is not for them? And maybe we can speculate of possible approach to overcome this. Thank you.
Yes. So we have not studied in great depth these few percent of T cell failures. I should add, and I said before they weren't technical, meaning that this is anecdotal, because 3% from 250 patients, that's not very many, right? That's 7 or 8, right? So it's still a small number. But in a few of those, we even had a second apheresis, and it still did not expand. So I think this technology, the way we use those beads and cytokine and media, is pretty efficient. And when it fails, it is usually because of the intrinsic quality of the patient's T cells. Now, we have not evaluated the underlying biology. It could be that these patients are highly deficient in a stem cell, a T cell memory component. That's a possibility, but we did not examine that. However, what I would say is these patients make the strongest case for these allogeneic approaches and ALL. So what can you do about it? Well, I think there are two things. One, you would try to understand it better and understand maybe how to fix that problem. Lots of ifs. The other one is go allogeneic. If your patient fails to expand, then go with an allogeneic T cell. You now have a justifiable reason for trying an allogeneic T cell.
Prежде всего, thank you very much for your excellent lecture. And the first, the most of us are pediatricians, and me too. So my question is, вы говорили о том, где можно и где используется эта возможность лечить с помощью КТ. Что касается лимфомы, меня интересует проблема неходжкинских лимфом. Вопрос вот какой. Неходжкинские лимфомы у детей — это совсем другая популяция, чем неходжкинские лимфомы у взрослых. Все, что вы говорили и было сделано у взрослых пациентов, на клетках, которые были у взрослых. Популяция Т и В-клеток взрослых. Есть ли что-то, что отличает детство, возможность использовать эту терапию принципиально отличает? Или это, в общем-то, просто другой поворот событий?
Michelle, Елена Владимировна is a key figure in pediatric lymphomas, non-Hodgkin lymphomas in Russia. So the question is whether, I would modify it slightly, whether among those non-Hodgkin lymphoma patients treated by today with CAR T cell therapy, are there any pediatric data, or do you think that pediatric non-Hodgkin lymphomas are different and we should expect or should not expect a difference in the CAR T therapy application to pediatric lymphomas?
Yeah. Unfortunately, I'm not aware of whether pediatric patients have been treated with Cd19 Car T cells. At our center, all lymphoma patients were adults. So I don't know. All I can say is that based on every knowledge that we have, there is no reason why those could not be treated. Let me add two things, two clinical points. In first protocols, disease in the central nervous system was an exclusion criteria at all centers, whether it's leukemia or lymphoma. However, there are now evidence in some patients with melanoma who received TIL cells, tumor-infiltrating lymphocytes, as well as in a few CAR patients, that T cells can traffic and eradicate CNS lymphoma. And so actually at Memorial we're planning a trial now specifically designed for CNS lymphoma in adults, which before, again, were excluded from all protocols. Secondly, I mentioned the neurotoxicity before. The neurotoxicity of CAR T cells is not understood. However, there have been protocols in glioblastoma, again, adults essentially with glioblastoma. And there the CAR T cells were infused stereotactically by the neurosurgeons directly in either fluid or tumor. And there is one case of a very spectacular complete remission obtained at the City of Hope in California. Other patients failed, but there was one very spectacular complete remission. And in that case, there was no neurotoxicity. And yet all the CAR T cells were put in the CNS, which still means that the neurotoxicity that I mentioned before is not dependent on T cells infiltrating the CNS. The mechanism is not well understood. But what it also means is that we should not be afraid of treating patients with CNS leukemia or lymphoma. And so they are now no longer going to be excluded from these protocols as they have been before. But I'm sorry that I don't know about the pediatric lymphoma. I'll just say that if they are CD19 positive, in principle, I see no reason why they would fail.
Thank you very much for a brilliant lecture. I think we are already convinced to use CAR T cells in our practice.
Thank you.
And my question is, what do you think, what kind of long-term effects can we expect from this therapy?
Great question again. That paper we published in February, I think is the longest follow-up of any study. It was the first ALL study in the US. And the patient the most far out is just a little over 6 years, and they're all adults. I think we need more data and more time, unfortunately, to have a good answer to this. But with respect to pediatric ALL, I would like to make the following point that I think is obvious to all of you, which is, chemotherapy can work, and it works in many patients. But I don't need to tell you about what the sequelae are, the developmental consequences of high-dose chemotherapy to children. I think we can say it's quite devastating. Patients are alive, which is wonderful, but they are not necessarily very healthy or productive citizens. And so the hope that we have, and I emphasize the word hope since we don't have the data, is that an immunotherapy will not have these developmental effects. We have this cytokine release syndrome and this acute transient neurotoxicity to deal with, but the long-term consequence of an immune response, all of this is a very violent, excessive, sometimes immune response. What would be the long-term effect of that in children? I certainly don't know, but I would also hope that it should be less than the long-term neurological and other and endocrine and whatever effects of chemotherapy. And therefore, this therapy should not be construed as just, what you need for 10% of patients who relapse, but possibly it may become a way to, in the future, eliminate chemotherapies if indeed the side effects are less.
I guess that's our dream, most of us. Any questions, вопросы?
Thank you very much for your terrific talk, and I have one simple question. So, what about the improvement according to the cohorts of patients? I mean, can you use it as a first-line therapy in any cases, or just for refractory patients only?
Yes. We think it should be used as a frontline therapy based on our recent analysis, but nobody has done that yet. The approval of these first CAR therapies it's not just that it's a historic moment because, a cell is approved as a drug. It's an engineered cell. It's an engineered cell with synthetic receptors. All of these are from a biological standpoint, first in their category. So there's a lot of historical significance. The way these CAR T cells were approved, though, is also remarkable in how it happened. It really happened very fast. I mean, yes, for me it's a lifetime. It started 25 years ago. But the approval was actually very fast. The first results in ALL were in 2013. First trials ever in the world. Not another antibody, not another small molecule, a completely new modality. And it was approved 5 years later. And the trials were phase 1 trials that were then expanded to phase 2 trials and eventually multi-center phase 2 trials. But two characteristics of these trials are that, number one, because they were focused on refractory relapsed patients, there was no alternative treatment. They are not phase 3 trials. They're not in thousands of patients. There's no control. In ALL relapse, what's your control group? It would be unethical. There's nothing. So they were not conventional phase 3 trials. Furthermore, the approvals were given with just, 90 or 100 or 120 patients, not, 2,000 patients. because of the urgent need for this patient population. So therefore, all the efforts of academics like us, and also the companies that got involved, was on these patients, and therefore the approvals are restricted to patients with relapsed refractory disease. We all agree that the data suggests that this should be implemented earlier, and we will see spends the money on doing now these frontline therapies. We want to do it academically at my center, but we have to find the financing for that. But we believe that's the logical next step.
Не вижу. Есть ли вопросы еще? Если нет, то, наверное, почти 2 часа. I think it's time to thank Michelle for this fantastic opportunity to contact directly and to communicate. Thank you so much for being here.
Thank you.
And actually, thank you for developing this for the patients, because it's been, most maybe of us here in this room are physicians, and it was always kind of personal and professional pain, a source of great pain for us to know that there is nothing else we can do for this particular patient. And they still come to us once, twice, 3 times a week, the patients whom there is no other chance for survival. So with the inventions you've made, for us it's another opportunity, like another big hope, and we all, most of us believe that this will really give a new dimension of care for these patients. So thank you for this.
Well, I want to thank you too. I want to thank you for the invitation. I want to thank you for your wonderful hospitality in the last 2 days. Thank you for giving us a tour of your facility. We were really impressed to see it's still a small laboratory, but it has all the right components to start the program. And we're so happy to see that you and your colleagues are interested in pursuing this approach. For me and for Dr. Riviere, this is the goal of a lifetime, to demonstrate that cells that have been reprogrammed and engineered can become powerful medicines. And it goes against the culture of the pharmaceutical industry, and it does not align with the culture of many hospitals. However, if we can show that we can succeed where drugs or radiation or sometimes transplants don't succeed, we think that we will generate the interest, the enthusiasm, and ultimately the financial support to develop a whole new approach to medicine, which is based again on an engineering of the patient's own cells. Again, I'm so happy that you've treated patients here. I think it's a remarkable tour de force that speaks a lot to your personal scientific and clinical and administrative abilities. I believe that this field will be driven by academic centers. It's wonderful that we have companies. We need companies to go large scale, to go global, and all of that. But there is so much to be discovered that we cannot rely on companies. Companies are here to give us medicines and also to make profit, and they have their own priorities. It's up to us in academia to really nurture and advance this field and to apply it to many more diseases. So I'm so thrilled that this center now in Russia is contributing to this global effort.